Protein labeling with stable isotopes are effective methods for quantitative proteome profiling using mass spectrometry. Stable isotope labeling by amino acids in cell culture or SILAC, which is a metabolic labeling strategy to encode whole cellular proteome, is widely used method for quantitative proteomics. In SILAC, two groups of cells are cultured in media that are identical in all the respects, except that one contains a heavy isotopic analog of an essential amino acid, while the other contains the normal light amino acid. After a number of cell divisions, the grown cells are combined and digested using trypsin. The complex protein mixture is further separated by SDS page to simplify the analysis. Further application is carried out by liquid chromatography, and purified peptide fragments are analyzed by MS. iTRAQ. It is a MS based technique for relative and absolute quantitation of protein. iTRAQ reagents are a set of 4 isobaric amine specific labeling reagents 114, 115, 116 and 117. An iTRAQ reagent consists of a reporter group, a balancer group and a peptide reactive group. Pooled samples are purified on a strong cation exchange as SCX column to remove the excess unbound reagent. These isobaric labels are detected upon fragmentation and release in mass spectrometry. The data obtained from mass spectrometry can be analyzed by using search engines such as Mascot. The analysis requires inputs regarding the experimental parameters such as enzyme cleavage, modifications, instruments used, peptide tolerance, etc. The data files generated from MS is uploaded and the search carried out by employing databases such as NCBI, MSDB and SwissProt. Now, let us talk about tandem mass tag or TMT. This method is similar to the iTRAQ which we just discussed. TMT is also MS based quantitative technique which uses the isotopomer labels referred as tandem mass tags. It also provides the accurate quantification of peptides and proteins. The tandem mass tags have been developed by the proteome sciences and currently commercialized by Thermo Fisher. I have given you the reference for the original study on tandem mass tag in the slide. So, these tandem mass tags, they are based on the similar principle of iTRAQ. Here the possibility for multiplexing is up to 6 possible labels. The TMT isobaric tagging technique can be used to perform absolute quantification by adding a stable isotope labeled internal standard peptides. It can be done by comparing the peptides from a target protein to a known amount of labeled standard peptide spiked into a sample. In that way, the absolute quantification can be obtained. The N-terminal amine and lysine residues are labeled through the NHS group. There are family of chemical tags which are based on the common structures. The series of TMT tags available: TMT 0, TMT 2-plex, TMT 6-plex, So, these TMTs are an innovative set of isobaric mass tags for labeling the proteins and peptides at amine functions, and mixing of up to 6 different protein samples are possible. While duplex and 6-plex labels TMT differ by the number of isotopic substitutions, the TMT 0 is the non-isotopically substituted structure that has been produced for only method development. During the MS-MS analysis, the TMT tags give rise to 6 reporter ions from 126 to 131 Dalton. Therefore, it allows for the relative quantitation. I am showing you one representative MS-MS spectrum of TMT labeled peptide, which is showing a reporter region. The relative abundance of target protein or peptide fragment in 6 different samples can be easily measured by comparing these signature mass peaks, which are generated by the different mass tags. Once the sample preparation is done, then further mass spectrometry analysis can be started. The peptides, which are differentially labeled, they can be mixed together and measured by the mass spectrometer. This method enables simultaneous identification and protein quantification. The labels react with the N terminus and reporter group is lost during the fragmentation. So, this method can be used to determine the relative abundance of selected peptides of interest from 4 or 8 samples. Therefore, high multiplexing can be obtained by using iTRAQ method. 4 independent reagents of same mass 145 can give rise to 4 unique reporter ions M by Z 114 to 117 in MS-MS analysis, and subsequently these reporter ions can be used for quantification of different samples. So, in iTRAQ, the quantitation occurs at the level of fragment ion spectrum at the MS-MS level. The peptides with the identical sequence, but derivatized with different isobaric reagents are indistinguishable during the MS spectra alone. That is where only during the MS-MS phase the quantitation can happen. So, it shows that identical fragment ion series for the peptide derived fragments, but it shows low mass reagent derived fragment ions that indicate that sample of origin and permit the quantitation. So, identification and quantification of peptides are achieved during the MS-MS analysis. The iTRAQ enables the quantitative protein profiling of multiplex samples without making MS spectra complicated. It reduces the redundancy to selecting MS precursor ions for collision induced dissociation and enhances the efficiency for MS-MS analysis and peptide identification. So, this is a representative spectra for the 4-plex iTRAQ experiment. The MS data is shown, and MS-MS spectrum is showing the reporter region, the signature of these 4-plex iTRAQ labeled peptides 114, 15, 16 and 17. Now, we will have a look on the 8-plex MS data. So, in MS-MS spectrum, now here we are showing the reporter region signature of a 8-plex iTRAQ reagent showing 113.14, 114.15, 115.15, 116.15, 117.16, 118.16, 119.16 and 121.16 reporter ions. Once iTRAQ experiment is completed, the data obtained from tandem mass spectrometry can be analyzed by using Mascot and other softwares. The MS/MS data analysis requires inputs from the user regarding the experimental parameters such as enzyme cleavage, modifications, instrument used, peptide charge, etc. The commonly used protein databases against which the MS information is processed to retrieve the sequence data include NCBI, MSDB and SysProt. The data file generated from the MS is uploaded and the search can be carried out. So, in this MS spectra, the pairs of chemically identical peptides of different stable isotopes can be differentiated due to their mass difference. The ratio of peak intensities for such peptide pairs demonstrate the population ratio for two proteins. So, here I have shown the light and heavy form separation with the 6 Dalton difference. The data obtained from tandem mass spectrometry following SILAC experiment can be analyzed by means of mascot search engines. The MS/MS data analysis shareware requires input from the user regarding the experimental parameters used such as enzyme cleavage, modifications, instrument used, peptide charge, etc. and the desired search criteria like taxonomy, peptide tolerance, etc. The commonly used protein databases against which the MS information is processed to retrieve sequence data include NCBI, MSDB and SysProt. The data file generated from MS is uploaded and the search is carried out.
Data visualization and qualitative analysis. Qualitative analysis. Click on the Agilent Qualitative Analysis software icon, which will pop up a new window. By clicking on Open a folder or file option, we can open a mass spec data file of interest. .d files can be opened. The chromatogram shows acquisition time period on x axis and the intensity of the eluted peptides ions on the y axis. Use right click of the mouse to zoom in the chromatogram. The bigger peaks which we can see are MS spectra. At the bottom of this window, we can see another window which shows different spectral peaks representing the precursor ions with the red color annotation on top of each peak. In the topmost window, from one MS spectrum to next MS spectrum, we can see many small peaks which represent MS/MS spectra. If the peptides are labeled with iTRAQ reagent, we can see the reporter ions in this MS/MS spectral view window. By clicking on the print option followed by acquisition method report, we can get the LC-MS/MS method which was used for the data acquisition. This acquisition method report contains details about method name, data acquisition mode, collision energy, gas temperature, flow rate, VCAP voltage, and solvent gradient details. We can save this file either in PDF or .xi format. Click on Export and you can convert the mass spec data file .d file into mascot generic file .mgf file, which can be used for analysis using mascot. Finally, we can close the file by clicking the folder icon given in the qualitative analysis window. Mass spectrometry has succeeded in identifying proteins based on the amino acid sequence information derived from the tryptic digested peptides. To analyze the data obtained from the mass spectrometry, sophisticated databases for protein identification were built, and PMF peptide mass fingerprinting, PFF peptide fragmenting fingerprinting, and MS ion search are the foremost. In the last 2 decades, many algorithms, new tools were developed to process the large data sets. Mascot is the widely used and trusted search engine for mass spectrometry data analysis. It has comprehensive database covering various organisms and is compatible with all existing mass spectrometry softwares. Besides Sequest, Xtendem is also used for MS data analysis. In addition to vendor-specific tools and databases, there are many online open source data analysis software. Apart from the mentioned softwares in the table, many open source softwares were introduced in the recent years, such as Trans-Proteomic Pipeline (TPP), OpenMS, MaxQuant, and many more. In the present lecture, we will focus on the iTRAQ data analysis using SpectrumMill software. SpectrumMill. SpectrumMill is standalone software used for LC-MS data analysis generated using Agilent LC-MS mass spectrometry. Here, we can see the home page of SpectrumMill having different tools for data analysis. Data extraction. LC-MS data analysis can be started using data extractor tool, where we can load our data files into the SpectrumMill and prepare files for MS analysis. Select the file from the folder named as MS-Data-SM folder and upload the selected file into the software. As we are analyzing the iTRAQ data analysis. So, before extracting the MS data, we need to assign the modifications on the peptide, carbamidomethylation on cysteine, and iTRAQ label on N-terminal amino acid and lysine. General MS merging constraints should be assigned as spectral similarity and RT and M by Z. Further, merge the MS2 and MS3 spectra from the same precursor. Keep the remaining parameters as default and save the method for future analysis. The extraction of MS files can be monitored in real time in right side, and once the extraction is done, we can proceed for the MS search. MS iron search is the tool used for data search against the database. Few important parameters has to be assigned for the analysis, such as database, species, enzyme used for digestion, missed cleavages, fixed modifications and variable modifications. First, we select the database as SwissProt. Enzyme as trypsin, species as Bacillus subtilis, and missed cleavages as 2. Fixed modifications were assigned as carbamidomethylation on cysteine and iTRAQ label on N-terminal amino acid. Oxidation of methionine as variable modification. Instrument is selected as Agilent ESI QTOF, masses as monoisotopic, precursor mass tolerance as 20 ppm and product mass tolerance as 50 ppm, which is quite good to proceed for data analysis. Once all the parameters are assigned, start search, and the search results can be monitored in the real time at the right side tab. Once the MS search is done, we can validate the data for statistically analysis using auto validation tab. Auto validation. In auto validation, both peptide and protein validation is needed to be performed. First, we will do the peptide validation by selecting peptide in Mode tab, 1% FDR, and remaining parameters are set as default. Click on Validate the files. Similarly, protein validation is done by selecting protein polishing in Mode tab. 1% FDR is assigned, 1% FDR is assigned, and remaining parameters are set as default. Now validate files. Protein peptide summary. Protein peptide summary is the tool to visualize the results obtained from the MS analysis. In the Mode tab, we have different options to display the results. We need to assign few parameters prior to displaying the results, such as keep valid in Filter Results tab to display only the statistically significant protein list. SPI was assigned as 60, which is an optimum value for MS analysis. In the review field, we can select species, molecular weight, score, peptide information, reporter ion intensity, accession number, and many other parameters. Keep remaining parameters as default and summarize the results. It will show the number of proteins identified or quantified from the analyzed sample. In the Coverage tab, the highlighted sequence will represent the peptides detected in mass spectrometry, and remaining sequences will appear in black color. It also displays the results with all the selected parameters such as protein name, species, unique peptides, score, and intensity of the reporter ion, accession number, and many more. Further, we can summarize the results based on the peptides, and we can export or save the results for further analysis.